So I think I will go ahead and introduce our next presenter. So Francois Botha from Synexa Life Sciences is our next presenter. And he and I believe some colleagues will be talking about leveraging LabKey for enhanced laboratory processes.
Thank you very much, Adam. Let me just quickly switch over to my presentation. Can you maybe just confirm that you can see it on your side?
That looks good.
Looks good.
Perfect. So thank you very much. I'm excited to share this with you all. We believe that we are using LabKey in a very niche way due to some complexities within our labs and our processes. So just before I start, just want to give you also an idea of who we are. So Synexa Life Sciences is a global leader in biomarker and bioanalytical services. And we essentially specialize in the development, validation, and delivery of complex custom designed assays. So our teams are spread over Cape Town, London, Berlin, Turku in Finland, Rockville in Maryland, the States. We have just over 100 experts that work in our science departments. And essentially they just help us they help provide innovative solutions to help our clients achieve their clinical and critical milestones. The type of work that we do spans from biomarker identification, development, clinical bioanalysis, and soluble biomarker analysis. And we use various advanced platforms like MSD, ELISA, RIA, fluorescence, luminescence-based technologies. But we also excel in areas like cell biology, including flow cytometry, ELISpot, and offer a few genomic services. So through close collaborations, we design these specialized assays and these specialized cases for a specific science. And as you can imagine, this broad spectrum of services that we offer basically requires us to have quite a vast system that allows us to be quite dynamic in the way that we use it, dynamic in the way that we use the data. So this is us. The presenting team today is myself. I'm the head of business information, so I take care of IT and business information systems. In LabKey's case specifically, it was more towards the governance side, the validation and operationalization. But then also part of this presentation, we have Chris van die Kerk, who's a business information manager, and Grant Peterson, who is our head of data management. So I wanted to share this with you just to give you an idea of the different challenges that we faced, and I tried to design this in a way that it goes from sort of like from the cradle to the grave. When you're thinking of when a sample's journey starts all the way to the end, where you report the data on it. So it goes through all the different steps. So the first one here is the complex data structures, where we essentially had really difficult time with managing diverse and dynamic data sets from multiple different sources, like e-manifests and Excel files from different centralized laboratories that all have their own ways of reporting or designing data structures. We also had some problems with flexibility of data imports. Essentially, it was really difficult in our previous manual system to handle diverse data formats like bulk imports and it required manual updates. And we had issues like overriding data. And it was obviously a very time-consuming process, which led to quite a few errors. And it also necessitated a lot of QC checks. We also had some troubles with bulk data management and updates. It basically just speaks to how vast our data sets are. And with a very manual system, it's really difficult to manage all those different data sets in a uniform manner. The next one on the list here is something that we didn't really think was a problem until we realized it was a problem. And it's essentially the advanced filtering and sorting of data. which was in our previous manual system. We had these conditional flags and formulas that essentially helped us to see the data differently. But it was not as intricate or as advanced as we really needed. So LabKey touched on that for us as well. User and permission management obviously was quite a big problem with manual systems because it was all file-based. We had problems with audit trail and compliance, as you can imagine, because the manual system didn't essentially have that. And all of this always also led to some data irregularities, validation problems, and it essentially caused a lot of problems with identifying irregularities and duplications and incorrect data formats in different spaces or in different areas of the data. So this definitely affected data quality at some point. And the last two on the list here is result tracking and data reporting, which was really difficult to report on these extremely large data sets holistically. And we definitely had a problem with scalability and performance, because basically what it meant was that we just needed more hands and more people and more eyes to help us with these QC checks in the various systems. So this is a good to give you a good idea of the challenges that we faced, and I hope that these next slides will give you an idea of how these challenges were met or solved. So I'll hand over now to Chris van die Kerk, who's basically going to be speaking about the sample manager, the use of sub-sample managers, and freezer management.
Thanks so much, Francois. So on managing complex data structures and the diverse sample data from multiple sample types and sources that we have, LabKey thankfully allows us to have multiple projects within multiple sample types each. This means that we can have complete separation of our different global projects, and our live and test environments can be kept separate, allowing for our CSA, testing, and validation environments each to have their own separate projects. Our global sample manager that you can see, the Synexa global sample manager, is where our global locations and freezers are set up. And each sponsor project has its own separate sample managers that are blinded to each other within the global sample manager, but that can share the same global storage space. Thanks. You can go to the next slide. That was me. There we go. So regarding our permissions management and managing our user roles and permissions across teams, we have quite strict user and group access control at the role level. Our users must undergo single sign-on and MFA authentications through the Microsoft Authenticator app. Within LabKey, we also utilize custom group creation to manage our groups on a site lab level basis, and we can nest our groups into supergroups where needed. We also assign different groups to different roles within projects, and all our projects are controlled at the individual study or sample manager level, allowing for granular control on how all of our permissions are defined. Next. Users are basically put into the groups, and the groups are assigned to our roles on a per project basis, allowing different groups or individual users to be assigned to different roles within projects. All while all of these projects are being controlled at the individual study or sample manager level. So we can have either individuals or users be part of the different roles. Thanks. You can go to the next. So regarding our freezer management and sample tracking, our global setup allows for shared freezer locations between projects. This means that our locations and freezers are set up globally, allowing multiple projects to utilize the same freezers while maintaining a shared storage management between the different projects. The setup and permissions for linking the freezers and projects works from both sides. So we can set which projects belong to a freezer from within a freezer, or we can set which freezers belong to a project from within the project. Next. The shared freezer locations between studies allows multiple freezers to utilize the same freezers. This allows for the complete separation or overlap of freezers between different projects. Here you can see that study 101 and 103 share none of the same locations or freezers, but study 201 has the locations of both of them. All at the same time, none of these studies are able to see or interact with each other's samples in the shared locations. Next. What's also great for us is that our users can only move samples to locations that the project has access to, so they can't move it anywhere else. If users move samples to locations that already have samples in them, then the system will move the samples around those occupied spaces and will prevent the locations from being double booked. Users are blinded from sample data from other studies and locations they don't have access to, or if they're in the incorrect project. And when our users check out samples, the freezer space is occupied as labeled and reserved. So our users aren't able to move samples to spaces where things have been checked out and are empty. With regards to entering samples manually or via a file upload, we can either do the sample setup via a header file that we have received from the sponsors, or we can do it manually if we have an idea, like if we're defining for the sponsors what the headers need to be and we're working with them, we can do that setup manually. Usually it's done via header files that we receive. What's quite nice for us is the wide range of field types that are available while doing the sample setup, especially regarding date, time, or any of the general restrictions you might require on data types. Samples are uploaded to our sample types, right, that we have individually in the different projects. Sample types are sometimes linked through SQL. We'll do this with, for instance, our barcode and scan lists that we have. And if we want sort of other fields, if we want matching on them, or if we want individual counting, we'll use SQL for that. So we've got our header files that help us set up the sample type. We have optional linking through our scripts. And the manifests, when they come along, we can import them through the sample imports and through the scan lists, so the different sample types. We can link those different sample types together. So thanks, Ron. So manual data entry is used mainly for the SRF forms that we still get, where manual entry is the only option. Other than that, we prefer to import from a manifest where we can pre-assign the field names and types and any aliases that might be required. I'd say roughly about 95% of our imports are done through the actual importing of manifests and not via manual entry. File imports also the option of also has the option of importing multiple sample types for us, which is quite useful, provided that the sample types listed in the manifest. Thanks. You can go to the next one. And so we utilize SQL to combine information from multiple sample managers, allowing analysts to scan samples into a separate sample manager from where manifests are uploaded, preventing our manifest area from being interfered with. We often want our manifest area where those are uploaded not to be edited in any way unless it's a correction from the sponsor. So we'll have two separate sample types. You can see in the example is study 401 serum Olink man and scan, man for manifest and scan for scan list. And they have fields that are being pulled through from each other that have been linked through aliases. So barcode man is the barcode in the manifest, barcode scan is the barcode from the scan list, and there's a calculation that happens to check if they're in each other's lists. If they are, then a match flag will be presented as scanned. Otherwise, the match flag will signal no match. For the example presented below, there was also a bit of a situation where the site would send through barcodes that had a site code attached to the last 3 digits or last 3 digits, but the samples themselves didn't have that site code on the barcode that was printed on them. So we had to make the code so that if it detects a match, even with the last 3 not matching, it'll pick it up as a scan sample. And that was quite nice. And you can see here we've got our counters. Counter man is counting each of those manifests in the barcode to let us know if there's duplicates. And counter scan is counting the amount of times this barcode appears in the scan list, and we can just see. So maybe there were multiple samples that had the same barcode, or maybe the sample was scanned twice by accident. Either way, these are sort of flags that we have to let them know. Next, I'm going to be handing over to my colleague to continue with the data section, Grant Peterson. Thanks so much, Francois.
Sorry. Hello, everyone. Thanks, Chris and Francois. Now we're getting to the exciting stuff, assays. Kidding. But yes, assays are more fun because there's just so much more to do. There's so much versatility. There's so many places you can safeguard your data and process it in different ways. So at Synexa, we run many different assays, as mentioned by Francois. And the raw data output from these machines vary a lot, from your neat, simple row by column data sets, where one sample is per row and its interpretation is there, to 96 well plate formats and data sets that are just completely scattered, and you really need to go and find out where the data is in that file before you can even use it. So LabKey luckily gives us the ability to change our data, to shape it into the right format for our scientists to actually see their data, and also for our clients. So as with sample manager creation, you can also set up your assay in that way manually, or you can enter fields and upload header files. And then there are, of course, a wide range of field types in the assay features, similar to the sample manager side, but we stick to using text. Why do we use the text field? Well, our assays spit out anything from numerical values, characters, special cases. So it's just become easier for us to convert them afterwards, either casting or using our scripts. Then onto the most important thing in my existence in LabKey is transform scripts. They are for pre-shaping your data before it even enters LabKey. Technically, it does enter LabKey because you're importing your assay, and then the transform script takes place to shape it into the format you want, and then it gets imported into the system. But I can't emphasize how amazing this feature is and how amazing it's been, because we're now using it for all of our assays, and these are run via R and Python. This decreases the load on the SQL, and if you know or don't know, SQL needs to reprocess each time you enter that specific web page. So having it run through the transform script, it basically leaves that data as static. So you're only loading what's already there. There is no recalculation taking place in front of you every single time. SQL is also not very friendly at all for data interpretation on a mass scale. And we do a mass scale, and yeah, we've had to shift and adjust, and it has worked beautifully. So with this in mind, obviously, raw data does not always show you something that may have gone wrong in the lab. So if someone happened to spill a sample or something, they would still get data, but obviously the interpretation of the data is going to be wrong, or it should not be included in a data set. So in this case, we do add extra fields into our assay to allow lab to add any other special cases, and then the SQL takes over at this point to override that final answer. But again, this becomes a very minor thing that needs to be processed, as opposed to counting 40,000 rows of data via SQL and then spitting out its final answer. Of course, there's auto linking for the samples in the sample manager, and it's very simple. If an assay is simple, sometimes your machine does take the LabKey sample ID, sometimes it doesn't, it can't take anything from the initial user. And then we use transform scripts. So through transform scripts, again, we are able to link the data and acquire the LabKey ID, even if it's not in the raw data file. This will be based on other data fields in the raw data file. So what will happen is we'll have a run ID column and a sample number inserted into our sample manager alongside the samples. The lab will then go and plan their analyses and put in, I'm going to run the sample in plate 1. It's going to be the first sample on my plate, and the raw data is going to say, this is plate 1, sample 1, please go and find my information from the sample log sheet, get my LabKey unique ID, and that then gets imported into the assay, which then pulls it all together and makes the system be aware that, oh, I am the sample that belongs here. Obviously, the lab needs to make sure that they're entering this information correctly. If a link is not found, luckily the system is going to kick that sample out, because if sample ID doesn't exist, it's just not going to import. And that raises flags to the lab to let them know, hey, you need to go and update your sample manager before you import this data. Otherwise, I'm not going to link to anything. Next slide, please. So to start off with, we're just going to chat a little bit about the Olink assay. It's a very simple, well, it's a complicated assay, but from a data perspective, it's very simple. Once you drag in your file, which is how we do this, it'll give you your top 3 rows. And obviously, keep in mind when you're using a T-script and your data is scattered, that header view doesn't really help much, but it's okay. For the Olink assay, everything is pretty much there. It's pre-processed, it's pre-calculated, nothing really special going on with him. Next slide, please. But the Olink platform has its own column in its own little processing machine called SID, which is unique in itself. And it's, like I mentioned, very easy, and you just drag and drop, and the sample IDs are lined up nicely in the sample manager section. One can then go and view all that sample's data by clicking on the blue hyperlink, and that's it. Straightforward, the easiest assay we have done thus far, but only 1 in 100 of the more complex things. So now we get to the real fun. Next slide, please. The flow module. So the flow module is a mega beast of an awesome machine that they have built somehow, I don't know, but it allows users to import their workspaces or their FCS files to access their data from their flow cytometry machines. We've opted for importing the workspaces as it holds pretty much the same information as the FCS files, and it would just be one import. So the results are extracted from the workspace in LabKey, and the general workflow is you import your workspace, and then you set up the whole product, and eventually we will get to the data transfer sheet creation. So that DTS step 8 there, we call it DTS, data transfer sheet, and that's the file that contains all the data that we will be sending to our clients at endgame. Next slide, please. So as I mentioned, in the beginning of a flow module, the lab will do all of their normal sample prep things and analyze it via Flojo. Something special to note, in Flojo, for the gating to be pulled out correctly in LabKey, it's got to be put into the statistics section. Otherwise, LabKey doesn't recognize it. Not a difficult step, hasn't caused us any issues. It's just something to note if you are working with flow cytometry data and LabKey. It's an extra step that your scientists will need to do. So they import the workspace, and then we proceed to going to the grid view of that analysis. We then activate all of the hidden fields under the statistics dropdown. That then gets exported into an Excel file, which is then decoded through a converter, which I built in Excel, into SQL ready language, SQL ready language. So these headers generally contain slashes and ampersands and other special characters, which doesn't bode well for the SQL. So what the decoder does is it really prepares it for, I can just go paste in my headers and then go throw all of that back into my query, my SQL query, to then extract my data. So that gets put into the first query. There are a bunch of queries in this study. And it will also extract any keywords that we need to bring and convert any fields which require conversions. Next slide, please. So for this study, just a little bit of an overview. One sample will, a sample will get pulled. They'll receive 2 aliquots, they'll get pulled, and these will then get split into 11 different conditions or stimulations. And each of these will be split into 110 parameters a piece, which will be used for further calculations. So I've been told that we're using 11 of our 13 lasers in our cytometers for this data set, which apparently in the flow world is like a big deal. I mean, it is a huge data set, so it must be really big. Anyways, to make life easier for the lab, we've put a few queries into play to flag things that will need their attention, because with the data set this large, they cannot be spot checking everything. They need to get the exact precise location of where something is going wrong. So the first query, flag stats, is your basic flow, query. We do it in all our studies. We did it before LabKey. It's basically if your frequency values are greater than or equal to 100, or if something turned out negative where it wasn't supposed to be negative. Flojo apparently gets overwhelmed sometimes and doesn't do its gating and statistical calculations properly. So we've added this as a web part query, which then also conditionally formats the fields for that line item which are damaged. And what's cool about the conditional formatting is you see it in LabKey in your browser or whatever, and once you export that data set, that formatting remains in your exported file. So there's no need for the lab to then go again and look for where is the damaged one. It's already highlighted to them in the file. They'll then go back to the lab and go regate and see what's going on. The next query is third query, metadata keyword flags. So those who know, the lab will enter keywords into their workspaces. This is basically information about the sample. And what this will do then is this query goes to check the sample manager side, and it checks the workspace to see, do I match up? Am I the sample that I'm supposed to be? Do I exist in the sample manager? And it also allows them to pull additional information from the sample manager that isn't in the workspace keyword section. Then also confirms that all the samples have been split across the required conditions. Has a condition been missed, or has a visit been missed where we expect there to be answers? So that's just a simple pull out the damaged ones only. If you're happy, you don't show me anything. If you're damaged, you're going to see I'm a damaged person. Please go and correct me. Then the fourth query is a viability duplicate count. This is just a little bit of extra thing that I had to build because at some point I noticed that my data transfer sheet was duplicating an entire sample, and it's because lab had entered the viability value of the pooled samples into multiple aliquots of that sample. So the way it was built is it looks for only one participant visit conglomerate concatenate should have an answer for viability, because you cannot have two viabilities for the same tube. And it then shows lab, look, you guys have captured this into two of the same tubes, essentially. Please go and correct it. Otherwise, this is wrong, and it just damages everything. So another simple thing, but that data also gets pulled into the data transfer sheet. So for any of the above queries, if anything is broken, the lab will go and fix the various issues in the various zones of either LabKey or Flojo, the sample manager. If it involves Flojo, the old workspace will be deleted in LabKey. They will go fix it and then re-import the workspace. Next slide, please. So now we've got another important step in this study, which is we call the expert and vaccine calls. So this is programmed via an R report, which identifies when a response from a biological perspective has been identified. And it's the lab's job to confirm that the calculated response is a correct response, or if it's not. So the lab will then get that file via the report, which will calculate the responses for either having an expert call or a vaccine response to something. They will then go check the gating, and then they will update their keyword that they've added to Flojo for the expert calls and the vaccine calls, and they will then re-import that workspace to override the pre-calculated answer. So obviously we can calculate these things, but we can't have the final say. We need the experts to decide for us, and that is the case with Flojo, where your answer depends on your gating. So the lab will need to go and check it, and this then brings out the proper answer for the client. So the final step, steps 8 and 9 here, that involves again using an R report to create our data transfer sheet. For this study, this has been our largest data sheet ever, really, as the way the client wants this data is in a vertical format, which where it currently stands is a 300 megabyte file spanning 200,000 rows of data across 28 columns. Now, some of you that might not seem like a lot, but for us, where we were dealing in Excel files before and having lab QC, that would be impossible. So this has just streamlined that entire process. It then writes that file into the files web part of LabKey for access to lab staff to go look at, data team can go look at it, and then we can send that file to our clients. Of course, as always, our priority is the safety of our data, and we literally cut out any manual data manipulation steps for the study except for lab correcting metadata. Next slide, please. And then just a little bit more on the data transfer sheet. So this R report for this study is targeting multiple zones. We're targeting the flow module, we're targeting the sample manager, and then we're bringing it all together in an R report. Obviously, it's first using queries, which one builds in the query browser in LabKey, because you have to have access to the data first in order to pull that access. And yes, then it goes to shape it, saves the file up there, and yeah. As Josh mentioned earlier, technology exists to serve people, and LabKey is doing a great job at that. That's all from me. Thank you. Francois.
Great. Thank you, Grant. Thank you, Chris, for going into the details of everything. Just a few last things that I'd like to touch on. The comprehensive audit trails, of course. Everything that you have seen in this presentation so far has been captured in the utmost detail in the audit trails, which can be configured for CFR 21 Part 11 compliant audit trails. So it meets those requirements, and it gives you surety and traceability of your data from the cradle to the grave. So just very short, I'm aware of our time, but as you can see from the solutions that were presented, essentially LabKey server has been able to answer all of these different or solve all of our different challenges and the different problems that we faced, from automating mundane functions, from removing multiple QC checks, from enabling us to create data files that were previously not possible just because of the sheer volume of data points. LabKey has transformed the way that we work with data, from where the sample enters the system all the way through to the archiving of that data, to safeguard it for future record keeping. It's an entire process. It's a closed process, and there's still a lot more that we can implement in LabKey that we just did not have time for in this presentation today. So that is all from my side. Just like to open if there's any questions, anything that anyone would like to know from us.
Yeah, there was, well, one question, since you've already sort of addressed the 10 major challenges and how LabKey helped with that. And I guess I'll say overall, what a great presentation. Really appreciate that and the work that went into it.
Thank you, Adam.
So I think there were a couple of questions that Chris answered directly in the chat. So maybe we're good with that. And just so that people hear, there was a question about importing assay data saved in a single file that needs to be linked to samples in different projects. And it sounds like that's not currently a use case for you. And then the other question was about essentially measuring the efficiency benefits. So as I say, Chris already answered that in the Q&A section if folks want to take a look at that.